exactly.
Right, but to get the point, so if I point
There's
it,
no way you can reach 100% of
I know, but I always
It's
want
over
to,
there.
you know, there's a part of me.
That's fair, you can send it directly to screen there.
yes.
All right, so we'll go ahead and get started. This is a session on image processing, and our first speaker will be Daniel Rowe from Marquette University.
All right, first I'd like to thank the organizers and being invited to this. So I haven't worked on image processing in a while, but I was asked to give a talk on this. So a long time ago when functional connectivity first became a thing, and I'm hoping everybody can hear me, when functional connectivity first became a thing,
You close the door back there.
that I had a lot of discussions with Bharat Biswal and like, is it real? Because there was a specific set of processing steps that you had to go through in order to see it. And if you didn't go through those specific sets of processing steps, you didn't see it. And I had looked at some single subject data and I didn't see it. So I started looking into the processing. And so here's an outline I want to talk about. So in fMRI, we know that a subject's placed in the machine and we divide up their brain into voxels, volume elements. And we want to observe, hopefully, or at least we imagine that we observe a volume at time 1 and a volume at time 2 and a volume at time 3 and so forth. And so these volume images are generally made up of slices or slabs. Okay? So I'm going to take a look at a single slab, and here I talk about, let's say for instance, it's slice 5. And so for this slice, let's say it's slice number 5 in this data set I'm going to illustrate later, then we have an EPI image at time 1 and time 2 and time 3 all the way up to time n. And this is usually while the subject is performing a task. So here's an example of block design. You would see an fMRI time series go up and down, for instance, for this designed experiment. And so for time series processing, what happens is you can take a time series of data, right? We'll start here at time 1 at the top and all the way down to time n. And so if you want to, say, smooth it to eliminate some noise or to have a low-pass filter, you can take this, say, vectorized version of this blue vector and multiply it by each row of this processing or filtering matrix. That in this example, I'm doing a 3-point average. And it will generate a red time series at the top from 1 at the top that's red all the way to n at the bottom that is less noisy. And so what happens is this process changes the statistical properties of our data. Okay? And so you can see that the mean is preserved. The mean is still about 50 from the way that I designed it. We made a histogram of these measurements, right? Right here it's just random noise to start with. Histogram of these measurements, mean of about 50, standard deviation of about, variance of about 16 by design. And so I also calculated the autocorrelation function for this time series, and you can see that the time measurements are correlated with themselves, but when you stagger them or take lags, there's really no correlation between the time series and shifting it by 1 or 2 or 3 or 4 and so forth. But when we've done this processing of, say, a 3-point simple binomial filter here, what happens is we, afterwards in red, we still have the same mean, but we have decreased our variance, of course, because we're averaging. We've decreased it down to a factor of the sum of the squares of the weights we have, which would reduce it from 16 in that example down to 6. I have the weights here. But what happens is now you have induced a correlation between the successive time points from the averaging. And here, this is the AR correlation, right, from shifts back in time, but it's also forward in time from this averaging. And this is a significant correlation here that's induced, 2/3 from the next time point. So what this tells us is we need to make sure that we account for these or get the data in the rawest form so you can do this or not do this and have minimally processed data. Okay? So we can theoretically evaluate this because we know statistics. So we know that if we have some vector V that has some mean delta and covariance matrix that I just called gamma, and if you're interested, I put all these slides on my webpage, they're on the front page. Then if we take our vector V and multiply it by some matrix O, such as this matrix that's doing the filtering, O, then we know how our mean has changed, and we know how our covariance has changed, right? Sigma. So then the sigma can, of course, be converted into a correlation matrix. And so we can examine the effects of this processing by, for example, saying, well, let's assume that gamma is identity. And so unless our operations are orthogonal, unless O is an orthogonal matrix, then we have induced some sort of correlation, which seems obvious but shocking. Okay? So let's assume that this gamma is identity, then we can calculate the effects of these processes. Okay? And so if we did the previous three-point filtering, and I have the wrong picture up here on the top right, then when we take that matrix that I showed you here a second ago, here on slide 6, and we multiply it by its transpose, and we turn that covariance matrix into a correlation matrix, then we can see what the correlation matrix is, and we can see that induced correlation, not just forward, but also backwards. And so, again, by simply trying to reduce the noise in our time series here, we've induced an AR, MA, and ARMA correlation through our data. Now, I want to show you an example with an image. So let's say that we have this image, a little smiley face on the right, as long as I'm on time, we have a nice smiley face. We can stack the rows of this into a vector, so we can take each row and we can stack them into a vector and multiply this vector of our image data, right? So we take the transpose of our image and vectorize it and multiply it by a matrix that might do, say, a local averaging in space. Now we're looking at spatial processing. And local averaging is space of our voxels to reduce spatial noise. Then that's going to give us a vector of image data that we can chop into bits and form it into a new image. And you can see that this smooth image on the left looks a lot like this original image on the right, but it's been smoothed, and so it's a little blurrier. So what I want to do is, whenever we have an image, we can number the voxels, and I'm going to number mine from top left to bottom right. Okay. And so we can stack the voxels into a vector, and we can calculate the correlation between these voxels once that we vectorize them. And then once we have this correlation matrix here in the top middle, then we can take a particular row of that corresponding to a voxel of interest, right? Particular row, so I have the 13th, the center one here. But because our processing is going to be the same for all voxels, it's not going to matter which one, then I can take this row of my correlation matrix and chop it up into the bits, right? And I can form it into a correlation image, and I can look at the correlation between my voxel of interest here, which is number 13, and all the other voxels. Okay. And all these slides are on my webpage for you. And so we can look at what the induced correlation is simply from doing a little local smoothing. And so this is one of the reasons why local correlation is pervasive in our data, because the scanner manufacturers know what's best for us. And so before the data comes off of the scanner, in reconstruction, they do lots of different smoothings. So if I look at my smiling face here, that's initially uncorrelated here, I can turn this correlation into a correlation image, right? So by doing the smooth, I can take the center row and I can turn it into a correlation if my voxels are initially uncorrelated with that identity correlation matrix. But if I do that smoothing, this is what happens, and this is the generated correlation matrix from multiplying, here on slide 14, this convolution matrix here on slide 11 by its transpose and turning it into a correlation matrix. Exactly the way described here on slide 9, where we can form a covariance matrix with that O convolution matrix and turn it into a correlation matrix. And so we can look at the effects of this smoothing, and we can see that we've induced a local correlation between the voxel of interest, which turns out to be this middle one, but it's the same correlation for any voxel that we choose, and the local neighbors. Okay. So looking at an actual brain image, this is a 96 by 96 image with 2.5 millimeter in-plane voxels, initially uncorrelated. So here's the correlation matrix. Zoom in, no correlation at all. If I smooth with 5 millimeter full width half max smoother, you can see that this is the induced correlation by multiplying the convolution matrix by its transpose and turning it into a correlation matrix. And you can see that I have a local correlation between my voxels. Okay. Now, but in reality, there's much, much more going on behind the scenes. Because in fMRI, the data is actually measured as spatial frequencies, or approximately spatial frequencies. And we generate our image, magnitude and phase usually thrown out, by the inverse Fourier transform. Now, if I think about a toy example, so here's an image much like the one that I showed you, the split between real and imaginary, and I take the inverse Fourier transform to get spatial frequencies, K-space. Then we can take this K-space and we can stack it, the rows, right? So we can stack the rows much like we stack the rows for that image, and we can do the inverse Fourier transform with a single inverse Fourier transform matrix, which will produce a vector image with real and imaginary that we can turn into magnitude and phase. Okay. Now, as I was alluding to, the scanner manufacturers and the physicists and the engineers that measure data know what's best for us. So when they take a vector of this K-space data, this is our mathematical representation, even though this isn't how they do it. We can take this vector of K-space data with real and imaginary, and so you can multiply it by a matrix that does any operations you want in K-space, your matrix that does your image reconstruction, and a matrix of operations in image space, like smoothing. In K-space, there's lots of operations to generate an image vector that we would turn into an actual image. They're split between real and imaginary and not turned into magnitude and phase yet. Okay? So a while ago, I looked at several different processes. So I looked at, here's an example of zero filling, apodization, which is a smoothing in K-space to get rid of Gibbs noising, inverse Fourier transform, which I've colorized here, just that same matrix, and smoothing. And when you concatenate them all together, here, of this series of matrices, you can see what they look like. They're all on the same color wash scale. And you can look at what the induced correlation is from this process. And so we can look at how it changes our image from the original image to how it blurs it, right? Make sure you look at your EPIs, the original images, magnitude and phase here. But also what happens is it induces this local correlation between your voxels from these different processes, right? From nothing all the way down to doing all those processes I described. And there's many, 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 many more. Now you're going to say, well, that's just a local correlation, why do I care? Well, it turns out that there are other processes like measuring less data where you skip lines of K-space and you got aliasing, you unfold. And so what happens is when you do the standard image reconstruction techniques like SENSE here with this acceleration factor of 3, and here's actual gel phantom and human experimental data, that you get correlation when you unfold the image and it's much larger in these regions when you smooth. Here's for sense. And it also turns out that this happens with grappa. So you can get these long-range correlations between different disparate regions within the brain. That, of course, we're going to say, oh, this region of the brain is correlated with this region. Right? So we need to be really careful about our statements. This effect was also seen in the Human Connectome Project data by my postdoc Ben Risk, who's now at Emory. We can concatenate these vectors for many different k-spaces of images and do this enormous same process to get to do k-space and image reconstruction and time series processing to get these image time series, and we can do much, much, much more. So in conclusion, time series and image processing changes the correlation structure in your data besides reducing noise. We don't get a free lunch, right? Some image reconstruction techniques generate long-range correlation. The simultaneous multi-slices through the planes. Here I showed you sense and grappa that's within the folds in a plane. We need to obtain a rawest form of data to see what it's like without any image processing, and with image processing, do the minimal amount of image processing possible, right? And of course, the topic of the next talk is to find ways to include or remove or account these image processing effects. So, thank you, everybody.
Thank you, Daniel. So we'll move on to our next speaker. We have Martin Lindquist from Johns Hopkins University.
No worries. Oh, that looks like me, yeah. Oh, thank you so much. And just the arrows and stuff. Yeah. All right. So I'm going to spend the first 10 minutes talking about this, uncovering interactions, and then the last 5 minutes I'm going to talk about some of the questions that Zhao stated in his email. And the title of my talk is about these uncovering interactions in these analysis pipelines. And so, wait, this is not working. Oh, there. And so it's very clear that, we've already talked about it a lot, that there's all these pipelines that are very difficult in fMRI data analysis. And that's not unique to fMRI, it's also in genomics and other things. And so traditionally these pipelines consist of sort of multiple distinct components that are put together and create. So you take a bunch of different software and you create a pipeline that takes you from the raw data to the final results, right? And so in this talk, I want to illustrate how sort of interactions between these different components of the pipeline can actually counteract some of the contributions of previous steps. I'm going to show you 2 examples of this. And the statistics here, I think, is pretty straightforward, but I do think it's a valuable lesson for statisticians working in these sort of pipeline-based fields. And so here's an example of the pipeline. You acquire your data, you reconstruct it, you pre-process it, you analyze it, you visualize it. But it's much more complicated than this, because if you go into the pre-processing, you have motion correction, co-registration, normalization, et cetera, et cetera, et cetera. And again, all these things are sort of, you pick and choose your favorite software and you build these pipelines together. And so oftentimes, the people who have constructed these little different parts don't actually talk to each other that much, and so it's sort of a leap of faith that all this is supposed to work together. And my claim is that it might not be so rosy. And so the first example here to show is just a very simple one, just to show you that this could be a problem. Is let's say that for fMRI data, you do some temporal filtering. You do some sort of filtering, temporal filtering, maybe bandpass filtering or low-pass filtering, and you do motion regression. Both of those things can be seen as projections, right? Because you can write them both as sort of linear regression models. And so my thinking, my claim is that if you perform, say, low-pass filtering after motion regression or vice versa, you can reintroduce the effects of one to the other. And that's pretty easy to see statistically, right? Because if we're doing motion regression, we're projecting it onto a space that's orthogonal to the motion. And then if we do another operation, we might move it out of that space, right? And so, and that turns out to be the case. And of course, this is addressable by, say, orthogonalizing the different steps with respect to one another or performing steps jointly or whatnot. So there are ways of doing this, but the fact of the matter is that most people don't do these things, right? And that's problematic. And does it matter? Yes, it matters. So for example, if I do a motion correction and then I do some low-pass filtering afterwards, you're reintroducing a correlation between the motion and the signal just by, changing the space that you're projecting onto. Now, this sounds, something that you can easily address. You can sort of motion correct you can sort of temporally filter your motion parameters, and this is fixed, right? So this is an easy thing to do. However, all these operations that we're building together, the data doesn't know that you've stopped pre-processing and suddenly started analyzing. So actually, the analysis can counteract your pre-processing as well. And so I was thinking about what would be an example of when you do filtering after motion correction. Well, we talked a lot about this already. What's dynamic connectivity? You do the sliding window, right? A sliding window is ostensibly a low-pass filter, right? So you're applying a low-pass filter after doing all this pre-processing. That's got to matter, right? And so the fact of the matter is, it does, right? So basically, you take your multivariate time series, you do a sliding window, and then they usually cluster the things to get these different states. They use like k-means or whatnot, right? And so this is sort of the standard thing. I think this is, from the Elena Allen papers, cited thousands of times, right? And so this is what most people do, right? And so my argument is that this act of doing a sliding window is ostensibly sort of filtering after you've done motion correction, and it's going to have impact, and it's going to introduce effects of motion, so, per Mark's previous point. Okay, and here you can kind of see it. It's basically you're applying a Toeplitz matrix to a signal, and that's sort of making you askew, right? And so you can see this with 2 as well. So this is bound to happen, and so I decided I would test this out. And so basically, I was a little naughty here. So I did motion correction here, and I motion corrected the images. But then I also saved the motion estimates and created motion images. So basically, this plus this is the original data, right? So this is what we don't want, and this is what we want, right? And I ran them both through the same process. Okay? And you want to know what happened? Got the same results. So these are the two states I get. This is doing motion, this is doing the motion corrected, and this is doing just doing the motion time series that you removed. That's no good. There's a million things that you could say. You could say that I was too aggressive in my motion correction, that I pulled some signal out, or I left motion there. But the fact of the matter is, it's no good that when the signal that I'm removing does the same as the signal that I'm cherishing, right? And so something is wrong here, and I think it's due to these weird interactions that we don't understand and that we need to dig into deeper. So this is something that I'm very worried about, and I think it's concerning for statisticians in particular, because we often take pre-processed data and apply our methods to them. Most of the time, you don't really know what was done to it, right? And so that's not a good thing. And that's why, as Hong Tu was saying, like, I agree with you, like, if you can do your own pre-processing, you at least know what was done to it, and you can know how they're interacting with each other. Otherwise, it's hard to. I mean, how? Is there any solution to us? Well, what about our AI overlords? Or are they going to help us? Well, they're promising to do all these things in one swoop. It's terrifying, right? Because you know what AI is? It's very unscrupulous. It'll do whatever it can to do better, right? It'll find some signal that it's not supposed to. And if it has to reintroduce motion to get better results, I'm pretty darn sure it will, right? And so this is not going to get better with these things. And I actually think this is going to be, these black box models are going to make this even worse, and we're going to have to guard against this. And so this is something that I'm personally kind of worried about. All right. So, what do we do about this? Well, one thing that I'm sort of arguing is I'm I'm against the sort of siloification of things. We're talking about we're doing statistical analysis, but we shouldn't be doing pre-processing. That's dangerous. Okay, we need to be involved with everything. We need to have at least have an understanding of everything and how it interacts. That's a lot of work, but that's going to be the best science, right? So I'm worried about siloification. You know, statisticians analyzing publicly available pre-clean data could be unaware of what was done previously, and this could have detrimental effects. And here's a little bit of a challenge, is that we really need a thorough statistical understanding of the entirety of the pipeline, and we really need a common language to talk about these issues. So I would really like to have some way of a statistical language of talking about every step that we do in the pre-processing pipeline, so that we can sort of figure out how the different steps talk together and how they interact. And this is, it's a very amorphous thing that I'm thinking about, but it feels like we have to figure out some sort of, you know, Dan has an example. He was, you know, you were working a lot with these matrices. That's a language, you know, stacking these matrix operations. I don't know if that's going to work throughout, but I think we have to think about that and understand this. And I think it's on us to sort of understand these interactions, because we're statisticians. We're supposed to understand variability. We're supposed to understand interactions. It's our job to help people who don't, right? And so I think that this is a grand challenge. I'm actually doing well on time. I just blew past. I blew past 18 slides there. And so these are some of the friends and things that helped me on this. Now, moving on to the next thing. So what are some of the most important research problems we should focus on in the next 5 years? Some of them are on this program. You know, the first one is, you know, large N studies. I mean, now we're having these epidemiological size, you know, 100,000 people studies, and those are really exciting, but that changed the way we do statistics, right? And how we think about results. Actually, another thing is small N studies. So there's these precision mapping studies where they put in a single subject for 20 hours and look at it. You should look at this work from University of Washington, Nicholas Dosenbach. You know, they've defined a whole new motor homunculus on like 3 subjects, right? It's amazing. Translational neuroscience, at some point we have to get stuff into the clinic, right? And so that's stuff that we have to keep working on, and that's important. I'm a big fan of brain stimulation because I'm a big fan of causality, and I think that that's one of the best ways to do causality in the brain. And then also exploring the use of AI. This could be like 500 different things, but some things AI is going to be really good for. I think segmentation is an example of something like that. You were talking about normalization. You know, recreating these algorithms in a more efficient way is probably very good at. Some of the other stuff like predicting depression, I don't think it's going to be very good at because we don't have enough data and it's too noisy. So where are the limits for AI and what are we doing? And so the next thing is, how can we, the statistical community, improve the processing and sharing of data set? And this is where I'm going to talk a little bit about the left turn my career has taken. I'm actually going to ask you guys for help here. So in the last 5 years or so, I've become a data collector. And so now I have a lot of data. So I'm one of the PIs on this thing called the Acute to Chronic Pain Signatures Program. And it's basically one of the biggest studies of the transition from acute to chronic pain. And so it turns out that between 20 and 50%, depending on surgery, of people that have an acute pain episode transition to chronic pain. And so the mechanisms driving this are really poorly understood. And so the NIH funded this Acute to Chronic Pain Signatures Program to sort of collect behavioral imaging, multi-omics, EHR data on close to 3,000 participants that were scheduled for surgery. So we have baseline measurements and all these things. They have their surgery, and then we follow them for 6 months, and we have longitudinal measures on these people. And the idea is that we want to identify biomarkers that show who is susceptible, using baseline data, who is susceptible to develop chronic pain versus who will be resilient to chronic pain. And then we want to understand the mechanisms of this because we want to understand, you know, should we change the treatment of different people. Well, right now, we have imaging data on 1,300 people. So that makes us a top 10 in the world imaging study. We're bigger than the HCP young adults. We just went past them. And 800 of those we have multiple time points on. And we're doing our first data release for public data, some 800 subjects, 600 with a total knee replacement and 200 with thoracic. And we want to dig into that, and we want to figure out cool stuff with it, and we want to use your methods. We have high-performance computing. We have people that can process the data. And if you guys have ideas and want to collaborate, that would be wonderful. So that's my challenge to you. I'm not done yet. and so this is the first release. So that's 800. The next release next year will be 1,400. And then it'll be like 2,000 up to the 2,800. So we're slowly building up for it. But if you guys want to be involved, if you want to try out your methods, then let's go for it. I also have a second data set. And this is a different one because this is 6 hours of scanning, so it's more of the precision mapping type thing on 101 subjects. So we have 2 hours of sort of naturalistic viewing. So naturalistic viewing is the new resting state. So it's basically people are watching movies or listening to stories while they're in the scanner. So it's sort of anchoring their connectivity vis-a-vis other people. We also have six different tasks that are in sort of the spectrum of different cognitive tasks, and this is also a rich data set. We're trying to look at, our goal with this study is to look at sort of individual variability and activations across this broad spectrum of activation. But if you have anything you want to do, any of these things you want to do, talk to us and we can get you some data and play around. And I'm even going to stop with 50 seconds to spare. You're welcome.
So we have time for a question. Yes, Daniel.
I just want to say that framework that I described, where you have a vector of all your original measurements on one side and you account for all the processing, things like image registration and motion correction, all of those can be included as matrices. And then in one big model, you can, if you have a big enough computer, look at what the effects are.
So when you talk about the students learning this pre-processing, and we've all struggled with it, and especially pushing students to do it. From your perspective, or if there is a biostatistics chair, do you see that some type of an integration of this pre-processing or some sort of a class that students want to go on the track of neuroimaging data analysis should be required in order for them to have a PhD with a focus in neuroimaging?
Well, I don't like requiring people to do things, but I do think that I think it's very artificial to say that this is statistics and this pre-processing is for the informaticians or whatever. It's all combined. It all matters. It's all a part of the same game. And if you don't think that, I think you're wrong. And so if I were a student now, I would very heavily dig into understanding the entire pipeline. It's going to make all the difference.
Thank you, Martin. So we'll move on to our next speaker, my colleague Heather Shappell from Wake Forest University School of Medicine.
Thank you. So I'm not talking so much about image processing, more about an analysis method. But first, I want to talk about Inside Out. Inside Out 2 is in theaters right now. Has anyone seen it?
Me.
Really? This many people have seen it? Don't give any spoilers. I haven't seen it yet, but I'm very excited about it.
They all die. No.
How many have seen the original Inside Out? Almost everyone. Who has no idea what I'm talking about? Okay, so several of you.
People that kids are too old for.
So, for those of you who don't know, Inside Out is a 2015 Disney Pixar film. One of the film's directors actually conceived of the idea while he was watching his teenager's personality change as she was growing up. And he and his co-director actually consulted with many neuroscientists and psychologists in an effort to accurately portray the mind. So just a quick synopsis here. This is Riley. She's the main character of the movie. She's an 11-year-old. And she moves from Minnesota to San Francisco because her dad got a new job in San Francisco. At first, she's really excited, but then she starts to get sad. She misses her old school, her old friends, her old hockey team. The other characters in the film are her 5 emotions, and these are living in her brain at a headquarters in her brain, and they control a command center there. And they dictate a lot of Riley's behavior. So for example, Joy here, when Joy is at the command center, Riley is happy. Fear takes over when he feels like someone needs to keep Riley safe. So for example, here Riley pauses right before running over the cord in this picture because fear quick grabbed the controls. Disgust keeps her from being poisoned, both physically and socially, but sometimes to her detriment, like here when her parents attempt to feed her broccoli and she flings it back on her dad's face. Okay? And then her dad tells her, you can't have dessert because she did that, and anger takes over. Anger helps keep her life fair. Okay? Anger takes over the controls, and now she is visibly angry and upset. And then the last emotion, sadness. Sadness actually doesn't know her purpose, and neither do the other emotions know sadness's purpose at the beginning of the film. So you have to watch it to see what sadness's purpose ends up being. But sadness takes over the controls, and Riley becomes sad. So Riley's memories in the film are actually housed in glass spheres here, known as memory orbs, and the orbs are colored by the emotional state that she was in at the time. And so in a way, when we're looking at these racks, we can see her state sequence that she was experiencing. And this sort of reminded me of what my colleagues and I are trying to do in our work. Ultimately, we want to estimate what someone's brain states are when they're in the fMRI scanner. And we won't actually be characterizing the states by emotion. Instead, we'll be characterizing them mostly by a network. So the nodes in our network are the different brain regions, and the edges represent high correlation between the bold activity at those regions. So here's an example from a study on weight loss in older adults. This is a study that took place at Wake Forest University, and it looked at the efficacy of 3 different treatment groups on weight loss in this older adult population. But what I became interested in was whether their fMRI scan at baseline, while they were being shown pictures of their favorite foods, and they're being told to imagine the taste, the smell, and imagine eating the food, right? Whether that could tell us something about what is going on in the brain that might be associated with whether they successfully lose 5% of their body weight at 6 months or not, regardless of what treatment group they're in. So for example here, this person, maybe they're in this blue network state, I'm just labeling it as blue, they see a slice of pizza and they transition to green, and then they go back to blue, but when they see ice cream, they go to orange, right? And we're interested in not just what the states are, but the actual state sequence and things like the transition probabilities. What is the probability you go from blue to green versus from blue to orange? And then the dwell time or sojourn time distributions, right? So what is the probability when they enter the blue state that they stay in there for a long time versus their probability of jumping out of it pretty quickly? So how can we go about doing this? Well, as Martin mentioned in his last talk, some people use the sliding window approach and then a clustering method like k-means clustering to group those networks into states. And Mark mentioned some cons of the sliding window approach, but there's other cons too, like what do you choose your window length to be, right? So other people have started using a standard hidden Markov model-based approach. This is becoming a little bit more popular now. And in this approach, the Y's here are observed bold signals at each GR, for the length of time that someone's in the scanner. We observe these. What we don't see is the actual underlying state that someone is in that gives rise to those bold signals. And these states represent unique brain networks of distinct activity and functional connectivity that repeat at different points in time. Okay, so people are transitioning in and out of these, and we want to estimate the transition probability matrices and the probability of the state that they start in, and then also here the Y's given the underlying states, that follows a normal distribution, we're saying. And that's parameterized by the mean and the covariance matrix of the state. So each state has its own mean and covariance matrix. And the covariance matrix is where we get our network from because you can convert covariance to correlation. What we don't like about this approach is that implicit in this model setup is that the time that someone spends dwelling in a state follows a geometric distribution. And what that's saying is, because the geometric distribution is decreasing, is that you have a high probability of only spending a couple of TRs in a state before you switch out of it. And that might not be true, and that could bias your results. So what we have proposed is using a hidden semi-Markov model-based approach, where I'm not going to go through this equation for the sake of time, but I want to highlight there's a sojourn distribution, the DUR, that is explicitly modeled here. So it doesn't have to be geometric, right? We can say it's Poisson, we can say it's gamma. We maybe don't know, and we can say it's non-parametric. And so it's right in our model, we can estimate it. And we use the EM algorithm actually to estimate these model parameters, the expectation maximization algorithm, to find the maximum likelihood estimate. So now we don't have that limitation anymore of that implicit model assumption of the geometric distribution. So what do? So we can fit this model to one participant if that person has a long enough scan time. Typically, though, we fit it on a group of participants, and we get a set of network states and the state transition probabilities and the dwell time distributions, right? But ultimately, we want to kind of compare these dynamics across different subgroups of people, like the people who successfully lost weight versus the people who didn't successfully lose weight. And so a lot of times we will fit it to the whole group, get our states, and then fix those states and refit the model on the subgroups we're interested in comparing, and get new estimates of the transition probabilities and the dwell time distributions with that set of states that we started with, so that you're kind of comparing apples to apples, right? So you can say this group spent more time in state 1 and this group spent more time in state 4, and you have the same set of states. The other thing is you can get a state sequence for every individual by using what is called the Viterbi algorithm, where you take these model parameters that you've estimated, you take an individual's observed data, and then you can say for each person at each time point in the scanner, what state were they most likely in? Now, we do have some simulation results showing that the hidden semi-Markov model outperforms the sliding window approach and the standard hidden Markov model when estimating the dynamic properties here, the transition probabilities and the dwell time distributions. But I don't have time to go over that today, but the results are published there, and I'm happy to chat about it after. I want to show you an actual application. So back to our weight loss example. Are there signature brain network states and patterns of state traversal that are associated with successful weight loss in older adults with obesity? And so we fit the model to a group of participants, right, all 58 of them, and each row here is a participant, and we fit 5 states to the model, and you can see they're transitioning in and out of these 5 states. But how do we make sense of this? So one thing we looked at is occupancy time, the total percentage of time they each spent in these various states, and then we took that, averaged it over the successful weight loss group and the unsuccessful group, and then compared. We did a permutation test to get these p-values. So what we saw was the unsuccessful group spent more time in state 2, and the successful group spent more time in state 1. So what were states 1 and 2? Well, we could represent our networks by a correlation matrix, but sometimes it's kind of hard to characterize the networks that way. So here we performed a modularity analysis, and this is state 1 and this is state 2 on our weighted networks. And what this means is any brain regions in the same color were more highly connected, right? Regions in different colors were less connected to each other. So what we saw was that in state 1, what we found most interesting is that the insula and limbic regions were more connected to each other, and the motor was off on its own. Whereas in state 2, where the unsuccessful group spent more time, the motor and insula were more connected and the limbic was off on its own. And so we're working with our exercise science collaborators and trying to tease this apart a little bit more. But the interesting thing is these 3 regions, motor, insula, and limbic, come up a lot in addiction and reward type behavior literature and obesity literature. So we feel we have something there. We also looked at sojourn distributions, but I'm going to kind of skip over this for now for the sake of time. But we did see differences in the dwell times, how long people were dwelling in these states between the 2 groups. And I'm not showing the transition probabilities, but we looked at those as well and saw differences there as well. So in my last 4 minutes, I just want to talk a little bit about challenges in future work pertaining to this method. I think the biggest challenge right now is how do we choose the number of states? And this isn't the only method that has this challenge, but when we fit this model, we have to specify the number of states in advance, right? And so far, the way we kind of do this is we try to fit kind of as many states as we can based on our sample size. There's not a clear sample size calculation, but we start to tell, like, if we fit too many, the results become unstable. And the other thing we like to do is look at the differences between the states, right? Like, kind of look at how similar they are. And if we see as we fit more, maybe two states become really similar, then we're like, okay, maybe we're overfitting. We really want kind of distinct states. But then that begs the question of, well, how do you define similarity? And then this gets into how do we calculate a distance between networks? So what we really need is a distance measure that will look at information levels. We'll look at the distance at 3 different information levels. There's low level, mesoscale level, and high level. And so low level pertains to information at the individual nodes and edges within the graph, right? You're just comparing nodes and edges to each other, but you're kind of ignoring all the topological properties. And that's the beauty of a network. The mesoscale looks at those topological properties and compares differences in things like path length or global efficiency, things like that. And then the high level looks at the spectral properties of the graph. Things like the eigenvalues and eigenvectors of the graph Laplacian, for example. So really, it would be awesome to have a distance measure that incorporates all of these. And we actually have a student in our lab right now. He's a PhD student in biomedical engineering, Mohammed Reza Kodeh, and he's working on this problem of coming up with a distance measure that will incorporate all 3 of these levels. So his distance measure will have the low level, the meso level, and the high level. He's looking at edge strength at the low level, mean shortest path at the meso level, and eigenvalues and eigenvectors of the graph Laplacian at the high level. And so that's currently work in progress, but there's still plenty to be done there. He's working on finding a way to actually figure out these weights here, the appropriate weights, because he's summing all these distance measures together. Another thing is, how can we best account for covariates in the model? Right now we're doing permutation testing, right, to do all of our testing, and it would be really nice to just have a covariate right in the model where we can test for group differences that way and also adjust for confounding factors. And it would be really awesome to be able to account for people who have multiple scans and account for that accordingly. So mixed effects, hidden semi-Markov model is something we have been working on, and I know, Mark, I think you've done some work on this as well. Yeah. The other thing, how can we account for larger network sizes, right? So right now, with smaller data sets that a lot of labs are working with, we have to tell them with their sample size, like, you can't really do more than 100 regions in your network. And that's a disappointment because they want to do whole brain analyses. Most of the atlases have 200 and some regions, right? And so they have to try to figure out what to include, or include subnetworks as their nodes in the network, and it would be really nice to just be able to do larger networks for this. So we're also working on a penalized version of the hidden semi-Markov model where we can shrink some of our covariance entries to 0 and allow for larger network sizes. And then lastly, as statisticians, we really need to make our methods available to the community if we want them to be used. And Mohammed Reza, the student I mentioned, has been working on a toolbox for this analysis and for the extensions that we're working on for this analysis, and that's hopefully going to be available very soon. So stay tuned if you're interested in running some of these analyses. And then here are my acknowledgements, the lab, people from the Empower study that I talked about, and then where this work all started was during my postdoc with Martin and Brian Caffo.
Thank you, Heather. And now we'll move on to our next speaker, Zhao from Indiana University.
First, I want to thank the organizer for inviting me to give the presentation. To be honest, I didn't do much work on data processing, but this is a framework we recently are working on in my collaboration with Brian Kaplan and Martin Lindquist and some other colleagues. So what we are trying to do is trying to utilize some like a property of the data and then try to skip some of the data processing steps. So basically when we do neuroimaging data analysis, the most of the time what we did is the so-called the localization analysis. So we're using functional residency fMRI as an example. So we collect fMRI scans, and then one step is to register those individual brains into a standard space, and then do parcellation, extract both signals, and then calculate the brain functional connectivity matrices, and then we can do either pairwise analysis or some like a network level analysis. So what we are trying to see at the beginning is that can we do a more global level analysis? So basically, if we assume the exchangeability in those functional connectivity, and then we can summarize the density function of those functional connectivity, and then use it as a marker, and then to do the analysis. And then later, we also can show that if we use the densities to do the analysis, we can actually skip the spatial registration step, and then directly work on data from the individual space. Just also to briefly mention some like a localization. So one issue in localization type of analysis is we repeat the analysis. For example, if we do pairwise analysis, we need to repeat the analysis for each pair. And then we always suffer the multiple testing issue. So what we did is we have a muscle level data analysis. We just trying to do a covariance regression and then try to identify brain subnetwork. And then we assume within the subnetwork we can associate with the covariance. So this is an example. So we identify some brain subnetwork associated with ApoE4 marker for AD subjects. And then later, even though we can do this muscle level, and then still it is not a global level analysis. So what we are trying to do next is, I was involved firstly in two projects leading by a former PhD student at Hopkins. So what we do is we consider density regression. So basically we assume exchangeability in those functional connectivity, and then we can then estimate the density of those functional connectivity, and then we can run some regression and study the property at a more global level. So basically, pretty straightforward. We consider a scalar on density regression at the beginning. So assume we have a binary outcome, and then we just regress on some like a covariance, scalar covariance, and the density function. But for density function, it is not in a Euclidean space. We cannot directly apply those functional data analysis approaches. Then we should consider some transformations, for example, log quantile transformation. And then we can just based on this estimation, we can just interpret the model and then at the odds ratio level. And then later, we also have a more general framework just to do a scalar on density regression. So basically, what we are trying to do is this is a more technical paper. So basically, we can show that by assuming some like a Gaussian process on those data and then some Bayesian approach, so we can basically skip the density estimation step and then directly apply the estimation on the discretized observed data and then to improve the estimation efficiency. So one of the theorem we show that the take-home message is that if the connectivity densities are invariant. So if we assume exchangeability and then use the connectivity density to do the analysis, this is invariant to those linear transformations. So basically, if we have a rotation on the brain, so then we will get the same density function. And then, so in this case, we can sort of skip the registration step and then directly work on the individual space. And then Brian and then his colleague, and they also have some like a recent work showing that if we work on individual space, we may gain more information about individual level information. So basically, if we register the brain into a standard space, so we may lose some like individual information. And then we apply the scalar on distribution approach to a transcurrent during current stimulation randomized trial on aphasia patients. So we reverse the analysis a little bit. So we have the binary treatment assignment as the outcome, and then we try to see if we can use the functional connectivity density distribution to predict the treatment assignment. So this is the result if we use the original density, basically ignore the property of the density functions and then just treating it as a function in the reproducing kernel Hilbert space. So then we couldn't find any association. This is the coefficient curve. So with the outcome, however, if we do a proper log punctile transformation, so we do see some association at the tail part of the distribution. So basically, if we, it is saying, so the tail part of the distribution of the density of the functional connectivity can actually distinguish between the two treatment group. And then if we look at into those tail functional connectivity, those are functional connectivity involved in the regions that was stimulated, so the inferior frontal gyrus. So this is promising because when we do pairwise analysis, so sometimes, so most of the time we may lose power and then we couldn't find any signal. However, if we do a more global level of analysis and then aggregate those signals together, we may potentially identify some interpretation there. And then later we consider another more general framework like a distribution regression. So basically, suppose we have both the predictor and the outcome are density functions, or we look at, for example, if we use structural connectivity to predict functional connectivity, what we usually do is just to do a either pairwise prediction or just try to use some like a statistical learning or machine learning approach to identify which pair or which groups of structural connectivity can predict functional connectivity. So this is also, so we look at a more global level and then summarize using the exchangeability in the connectivities and then try to do a density on density regression. So basically we can summarize the density from the structural connectivity and then also the density from the functional connectivity and then try to see if there's any association between the two and then whether the association may differ across different population. So when we have this, this is our proposed model. So we have this because the density functions are not in the Euclidean space, and then we try to find a proper geometric we try to find a proper Riemannian manifold, and then do all the analysis on that Riemannian manifold. So what we did is like we use the half density functions. So basically, this is PI is the for example, think about the regression analysis. This PI is the point on the regression line, and then we consider arrow term, and then also project back to the half density, and then to get the regression model. So this is a geometric interpretation. So then we have a PI, which is the point on the regression line, and then it lies in the half density sphere. And then we have the tangent space induced by this point, and then we define the arrow term in this tangent space, and then we project back to the half density space. Then that will be our observed outcome. So that's basically what we are trying to do here. We have this density on density regression framework. So these are all the, and then we try to connect the two using a Wapping function. So this Wapping function is you can think about it as an analogy to the beta coefficient in linear regression. So we use this Wapping function to give a more straightforward interpretation about how those two densities are connected or transformed from one to the other. So this gives an example of how those two are connected. So for example, I started with the density functions, and then based on the shape of the Wapping function, so we can see the density is either skewed to the right, skewed to the left, or skewed to the right, or changing the variance of the data. So, and then if we can just have an estimate of the Wapping function and then look at the shape, so then we can know how those two densities are connected to each other. And then again, the Wapping, to estimate the Wapping function, so basically this is all from the, I'm not going into too much of the technical details, but it's all based on the Riemannian geometrics, and then we can transform those into the L2 space, and then define the metric, distance metric, and then also have the propositions and then theorems to show the consistency of the estimators, and then also give the inference. So here, again, so for beta estimation, so the wrapping function is monotonely increasing and then with the boundary condition. It is not in a linear space either. So then we have to do a transformation on the wrapping function and then to get the S, and then you through this homogeneous linear differential equation, then this W weight function is actually in the reproducing kernel Hilbert space, and then we can estimate that first and then transform back to the wrapping function. And then we also provide the inference on the wrapping function estimation. So then we go to HCP aging study. So we have the density functions from structural connectivity of DTI, and then also the functional connectivity from resting state fMRI. So this is the DTI function, structural connectivity is the predictor, and then the resting state fMRI density is the outcome. And then we look at the data and then split into two age group by the 65 years old cutting point, and then also feed the model to male and female separately, and then to see if there's any difference. Also, I think in Tintin's study, they show some like a difference between male and then female, and then year pattern, age pattern. So this is also what we see here. So for example, if we use the predictor using the number of fiber bundles, so we know the distribution of the number of fiber bundles is super skewed to the right. So then we see this convex curve of the wrapping function because the functional connectivity is distribution is closer to the bell shape. So we see the difference. Sorry, the red curve is the female, and then the blue curve is the male, and then the solid line is less than 65 years old, and the dashed line is over 65. So we see difference between male and female, and then also between the two age group in this. So basically, we can say that, so when we look at the association between structural connectivity and the functional connectivity, there is difference between those. And then also, if you look at the median FA and MD, so those two, median FA is closer to a bell shape. So then we don't see, much difference at the, so we see some difference in the tail range, and then also the same for the median MD. So median MD is also like a skew to the right distribution. So just to summarize, so we consider a density regression approach to try to avoid some spatial registration step. And then because we believe that if we can properly skip this step and then may gain more individual level information, and then this also provide a more global level analysis rather than localized association, and this may potentially apply to non-registered data. And then thank you. And then these are my collaborators, Zhao, who lead the first two scalar on density regression approach, and then Brian, Abi, and then Martin, and then Vadim are from the biostats department, and then Kiana is the PI for the aphasia study. And then thank you so much.
So we have time for a question for you before we open it up.
So for the two, so you had scalar on distribution regression and then density on density. Second part you use the square root, the first part you use the log. Why not use the same for both, or which one is better? You can use both in both situations.
So for the second part, because we have the predictor and the outcome are both density functions, so they are in the same Riemannian manifold space. But for the first part, the scalar on density, so one is in the Euclidean space and then one is on the in the manifold. So it's a little bit harder to link between the two.
I think you can do either in either case. It's a matter of
right. So
information.
right. So we have to. So for if we do a, so I think Hong2 also have some like a geodesic regression and then also other type of work just directly dealing like a scalar on manifold and a manifold on scalar type of regression. So and then I think there's also some recent work just utilizing Worcester regression type of framework to do that. Yeah, I think that's doable. But for us, I think we specialize the density on density regression just because we have densities on both sides.
Thank you. And now we'll open it up for questions for all of our speakers.
I still have question to you.
Oh.
So you mentioned that this
something? You think all the connections are from the same distribution? So how to understand that?
So basically, so suppose if we linear rotate the brain or change the labels, we still get the same distribution. So that's the exchangeability assumption.
Do you mean that the connection are not region specific?
So it is. But when you, so for example, if you label them if you change the one column of the labels, you still get the same distribution. That's exchangeability.
This is from Martin and Dan. So there's been literally thousands of papers with questioning state connectivity, and you seem to be showing that there might be some artifacts that are not really about how the brain works. How worried are you that these results are do you think these are really biasing the whole field, or are these what order of magnitude should we be worried about this?
I mean, I don't think that you should throw out a whole field. That's not the. I don't think that's the argument that I want to make. I mean, because it's clearly that you could take resting state data and predict things that are meaningful, and there's a lot of meaningful results that you can get, right? That said, I'm worried about how sort of non-neuronal information is influencing that. I mean, it could be that motion is predictive of these things, and then you don't really need $1 million scanner for that. You can just sort of look at how much
motion predicting ADHD, right?
Yeah, exactly. I mean, you don't need a brain scanner to diagnose ADHD. You just see if they're under the table after 2 minutes, right? So I mean, I think that I'm more worried about those things. But I'm not. I just want us to be careful. And I also think as statisticians, it's our job to be careful. Sometimes we're the wet blanket, right? But that's okay, right?
Other questions? Yeah. So I have a question for both Martin and Yi. So you show the use of the different pre-processing in the pipeline can have a bigger impact on the subsequent statistical analysis. But for you, he said if you pick the right statistical approach, the result can be invariant. It's not up to the the processing step. So the reproducibility has been a bigger concern in neuroimaging community. So and it looks like really up to the statistical approach being used. Do you have any comments?
I think one of the cool things about Yi's work is that it works on sort of the native space and it circumvents one of these big things. So I mean the crazy thing is that you take everyone's brain and you warp them onto a standard thing. That seems like terribly invasive. And Yi's work is cool because it sort of circumvents that. And so I think the exploration is figuring out what's won and lost by doing that. That's why I think it's cool. I mean, do you have anything else?
So I still, it's like a more global level. So we are still working on how promising the promises how this may provide more insights into those scientific implication. So I think even though after that, so we know there's signal there, but still sometimes when I talk to collaborators, they want interpretations, and then which are the brain regions actually. So sometimes we still need to go into those localizations. So and then if we still go into localization, then the next question is, if we skip this step, is it really skip the right thing? So that's another question.
Other questions?
Yeah, I have a question for Heather. So you study this kind of weight loss for overweight. I wonder how you take into account the cardiovascular factors because they affect the both signal or not.
Yes, we did not take that into account really, unless that was removed in some pre-processing step. So we don't have it built into the model at all.
But do you see the differences in those factors between those subjects?
Well,
like blood pressure,
we haven't really looked at that. There was one toy example we did. Do you remember this when we started with the model and we looked at how heart rate was correlated to to brain regions in a high anxiety task.
Oh, yeah.
And it was very correlated.
Any other questions? Yes, Fernando.
I have a question for Heather. Very nice work on the regime switching. So the way I see it is that at every time point, there is a really unique regime. But with this approach, you're sort of discretizing that, right? You mentioned about different distance metrics
Right.
in order to come up with the right number, the best number of states. Now my question is, how do you do this across many subjects?
Yeah, because we're fitting one set of states for, but you're saying it might differ by subject, right? Yeah. It would be neat if somehow in our mixed effects hidden semi-Markov model that we're developing, that we can have some sort of random effect in there that says how much a subject deviates from these networks that we're fitting to the whole group.
So those five are like your basis functions common to all of them, and then
Right.
there's a bit of a deviation for each
Right.
of them.
Yeah.
Thank you.
Because as a comment on the number of states, I was just thinking that there is a whole number of methods in statistics that tries to estimate the number of states, and that maybe can be something that you want to look at. Basically putting a trial of the number of states, also the weights of the mixtures typically, they can be seen as, they can be made dependent on distances, for example, or other things. So it's going to be more complicated computationally, but I think that's probably something that you may want to look at.
Yeah. So once we have our distance measured, to go that route.
You can, yeah.
Thanks.
Other questions? All right, let's thank our speakers again.